If you've ever worked in biology, you know the feeling, right? You run a huge experiment and you end up with this massive, overwhelming list of genes. It's a fantastic result, for sure, but how do you find the real story that's hiding inside all that data? Well, that is the exact problem we are going to solve right now. So just imagine this for a second. You've just compared cancer cells to healthy cells, and your analysis spits out a list of 1,000 differentially expressed genes. All that means is these are genes whose activity is way different between the two groups. That's great, but a list is just a list. To find the actual biological insight, we have to ask a much better question. And this is where enrichment analysis comes in to save the day. It's basically a computational method that's built to answer one simple question. In my big list of genes, are there certain biological functions or pathways that show up way more often than you'd expect just by random chance? To really appreciate why this method is such a game changer, let's first dig into the fundamental problem with just staring at a raw list of genes. See, the goal here is to shift our thinking completely, from individual genes to broad biological themes. We stop asking which specific genes changed, and we start asking what kinds of genes changed. Like, are the genes that got turned up all related to the immune response? And are the ones that got shut down all involved in the cell cycle? It's all about finding that high-level story. So how do we actually do this? Well, like any good analysis, getting a result that actually means something depends entirely on setting up the data correctly right from the very beginning. Here's a quick roadmap of the whole process, from that initial gene list all the way to the final story. We're going to walk through these 6 key steps that take you from just raw data to real, actionable biological insight. Okay, first things first, you need your list. This comes from what's called differential expression analysis. You use statistical tools, a really popular one is an R package called Lemma, to find which genes show a truly significant change between your groups. This list is the foundation for everything that comes next. And let me be clear, this is absolutely non-negotiable. You can't go looking for patterns in a list of significant genes until you have a statistically solid list to begin with. You've heard it before, garbage in, garbage out. Next up, a bit of critical housekeeping. Gene names can be kind of messy. Sometimes they're ambiguous or just plain outdated. To talk to all the big biological databases out there, you need a stable, universal language. And that's where entries IDs come in. Think of them like a unique social security number for every single gene, making sure you're always talking about the right one. All right, now we get to a really critical idea, the gene universe. To say a pathway is overrepresented, you need something to compare it to, right? But that baseline isn't all 20,000 plus genes in the human genome. Your universe is only the collection of genes that you could have possibly detected in their specific experiment. For instance, every gene that was on the microarray chip you used. Getting this background set right is absolutely crucial, because if you get it wrong, your statistics will be biased from the start. This next step is so important. We don't just analyze all our significant genes in one big bucket. We split them. The upregulated genes, the ones that got more active, often tell a completely different biological story than the downregulated genes. Analyzing them separately is essential if you want to see the full picture. All right, our data's clean, it's organized, it's ready to go. Now we get to the heart of it all, running the actual enrichment test. Now, the classic method is called overrepresentation analysis. It basically uses a statistical test to ask, hey, does this biological pathway have more of my significant genes than I'd expect just by random chance? But today, most of us are using modern packages like Cluster Profiler, which runs in R. These tools are way more sophisticated, and most importantly, they're designed to create the awesome visualizations we need to actually see the story. So the computer has done its job, the numbers are crunched. Now comes the fun part, and honestly, the most important one, turning that raw output into a compelling biological story. What you get back from the software is, well, a big table. It's a list of biological processes, pathways, and a bunch of stats like p-values. It's incredibly valuable for sure, but it's not exactly a page-turner. So how do we translate this into something we can actually understand and communicate to others? The answer, 9 times out of 10, is a killer visualization. And for enrichment analysis, the undisputed champion of the world is the dot plot. A dot plot is just so effective because it packs in a ton of information. Each row, that's a different biological pathway. The size of the dot tells you how many of your genes are in that pathway. Bigger dot, more genes. And
The color, which usually goes from cool to warm, shows you the statistical significance. Basically, how likely is it that this happened by chance? So, in a single glance, you can spot the biggest, most significant stories jumping right out of your data.
So, after going through this entire process, what are the most important things to remember? Let's just boil it all down to the key takeaways. Okay, the single most important thing to get is this. Enrichment analysis is not about focusing on one single interesting gene. It's about zooming out to see the big, coordinated biological trends that are driving the changes in your experiment. It's the forest, not the trees. That's right, you've successfully shifted your perspective from the individual actors on the stage to understanding the title of the play itself. It is all about the big picture. And please, never forget this. The genes that are turned up and the genes that are turned down can represent fundamentally different processes. A cell turning on its immune defenses while shutting down cell division, those are two distinct, super important stories that you'd completely miss if you just lumped them all together. And finally, this whole process is the step that actually gives your experiment meaning. A list of 1,000 genes is just data. But knowing that those genes are overwhelmingly involved in something like T-cell activation, that's knowledge, that's the story. So the next time you get that huge spreadsheet of results, don't just see a list of genes. Ask yourself, what's the real story here? What hidden biological narrative is just waiting inside your data, ready to be discovered?